Okay, so the first question that you are faced with is to look at these plasmid maps and to determine when you digest them with these two restriction enzymes, which are BAMH1 and HIND3, what size the linear fragments will be. So if you remember, if you recall, when you cut them, we'll start with this one here, the pecan R. You would cut them at this site with the HIND3 and this site with the BAMH1. This will leave you with two fragments. One of the fragments is this really large fragment here, and the other fragment is this one in the middle. This fragment, it tells you, is 807 base pairs long here. So all you need to do to work out the size of the two remaining fragments would be take the whole number, so 5, 5, 1, 2 base pairs, and subtract 807 base pairs from it. That should give you 4705 for the large fragment here around the outside, and it already tells you what the 807 is. Doing the same thing here by subtracting this little bit of DNA, which we're going to cut out, which is 377 base pairs from this, so 4872, which is the whole thing, minus 377, should give you a fragment of 4495 base pairs. It's as simple as that. Okay, you're cutting out these sections, and you're leaving one larger section and two smaller sections. And these are what the linear fragments will look like when you've actually taken them out of the gene. If I flick back to this, you can see where you started with, you can see where they're cut, and you can see what you would end up with here. Now, at this stage, you are asked for the next question, drawing out these linear fragments, so you can check, if you pause this here, you can check to see whether you got the linear fragments right. If you got the linear fragments right and you're struggling with these restriction sequences and leaving the sticky ends for the next question, I'm pretty happy with that. This next bit gets a little confusing, and it's probably only if you're really pushing for the very top grades that you need to concern yourself with this. But knowing how restriction enzymes work is quite important. So we've got two restriction enzymes here. We have this BAMH1 enzyme and a HIND3 that we're going to use, and these are the sites that they will recognise on the DNA. And these arrows here, they show you where it would cut the DNA. So here, and it would cut here as well, and it would leave this molecule here with this on the end. So if I shade this one in green to show you, so you'd have this, if this was a big bit of DNA, would end up with that sticky end, and then there would be another piece of DNA that it was joined to because you've cut it, so this is the other piece of DNA that would be left with this sticky end. Okay, so that's how they work, and then you pull them apart and it leaves these two sticky ends here where those bases are. And they cut at different sites, and what it asks you to do is to line up those sequences and to add on the correct sticky ends, and it's really tricky. So again, these are just some examples of how these enzymes work, these restriction enzymes. So this is a HIND3 enzyme, and you can see it's cut here, it will also cut here, and that will leave you with protruding ends looking like this. Again, here you've got another enzyme that will do a digestion, a different one, and it will leave like this. These are called sticky ends. Some of these restriction enzymes will cut straight across, and they're not quite so useful, It's much harder to stick things back together when there's no sticky ends for us to use, and it will leave you with blunt ends. So for your course, the thing to remember is sticky ends, leaving sticky ends, using restriction enzymes, cutting the plasmid and cutting the gene out with the same restriction enzymes which leave complementary sticky ends. And if you remember that, you're going to be pretty good. As I say, this next stage is much harder. So here we've got our pecan R plasmid, which for some reason has decided to chop a bit of it off, but I'll just draw it back in as a circle. And here in this section we would have had the HIND3 section and the BAMH1, so this would have been the section in here. This would have been the bit that was cut by HIND3, and then you've got the BAMH1 bit here. You would have had the okay. So you would have on this plasmid, these are the two fragments that you would get. So putting the sticky ends on is the bit that becomes very tricky. The way that these would read is that this would read around here from 5' to 3' around this way for the top strand, and the other strand would be going the other way here from 3' to 5'. So the clockwise on the outside is 5 to 3 and the inside is 3 to 5. If you look at this and you look at this fragment that's been taken off here, you can see if you stretch this up that this would be the 5' end here, so it would be 3' at the bottom and 3' here and 5' at the bottom. This picture at the bottom that you've got now is probably the only thing that you might be faced with in an examination where you have to label sticky ends. So let's go up here and let's have a look at the site we've got. Let's try this end over here first, so the HIND3. So where does HIND3, what does it do at the end? Okay, so we're going from 5 to 3', which is this bit here. So it will cut here and it will leave a sticky end on the end looking like that and this. So this one here is going from 3 to 5', so this is the bottom strand. So on the bottom strand we would have just an A, and on the top strand we would have a T, T, C, G, and A. And normally in any description like this it would be reading 5 to 3' in this direction. So remember this is 5' to 3' here in the top strand. And then we've got our BAMH1. So on our 5', which is the top strand, we would be cutting like this, and it would also cut like this. So it would leave us with a G at the top on its own, and then a C, C, T, A, and a G there. And there, that would be the sticky ends that you'd have. Now, remember I said that clockwise here is going from 5 to 3, 5 to 3' in this direction for the top strand. Well, our PBAD gene here is going to be going 5 at the bottom if you look, so this would be the 5' end at the bottom to 3' and 5' at the top. sorry. It would be going from 5 to 3, so this is the 5 end, this is the 3 end. Oh, sorry. So you're coming around here, this is our gene, this is the 5' and this is the 3', but this is on top. So 5 and 3 here at the bottom. Five prime and 3', and then at the top it must be going anti-clockwise. So we would have 3 to 5, so the opposite in this direction. So 5 to 3, and then 3 to 5. So what would the basis look on this one? So 5 to 3, BAM H1, BAM H1, 5 and 3. So again we're going to have a G, and then a C, C, T, A, G. And then over here we would have a, for this one, the Hind 3, you're going to have a T, T, C, G, A, and then at the bottom just an A. Okay, so that's for those ones. For the next sequence that we have here, we've got these two here, and again this will be the same as the last one. So we have our 5' up here and 3' up here, and then it would be going the other way, 3' to 5' at the bottom. And the same here, if we come round to the BAM site here, this is 5 and this is 3 to the Hindi site. So BAM to Hindi, 5 to 3 at the bottom. And at the top it would be the opposite way around, 5' to 3'. So again we just look at these restriction enzymes and we can see for the BAM H1 on this end that we'll be left with a bit like this. So for the top bit will have a G, and the bottom bit would have a C, C, T, A, G. And over here on this one, for the Hind 3, we would have a bit like this. And so we would have a T, T, C, G, A, and at the bottom just an A. And on the BAM H1 bit, it would be it would leave you with a G and a C and a C, T, A, G. And these bits here are the sticky ends, here and here, and well, the one's gone from up here, but you can see, you get the picture. So if you struggle with that, just don't worry about it so much for now. But that's how it works. Okay, so it says now for question 3, in order to create a plasmid that can produce the red fluorescent protein in bacteria, what components are needed in the plasmid? So you would need a sequence to initiate the DNA replication. So this is an ORI gene, which is that would initiate replication. Very important. You would also need a promoter sequence for initiating the transcription of the gene. So you would need a promoter for the gene you added. So in our case, that was P bad. you would need your gene of interest. So in our case, that was the red fluorescing protein gene. And you would need an antibiotic resistance gene. And that allows you to select just the bacteria with the plasmid in them. Because what you can do when you've got the bacteria is if you, for example, have the ampicillin resistance gene, you can expose all the bacteria to ampicillin and only the ones with the plasmid, because it has the ampicillin resistance gene, will survive. So it's a way of selecting. It's really clever. Okay, so 4, bacteria can be killed by antibiotics unless they carry a plasmid that has the gene for resistance for that antibiotic. Biotechnologists call these genes selectable markers because only bacteria that carry the gene will survive an antibiotic. If the uptake of DNA by bacteria is inefficient, why is a selectable marker critical for cloning a gene in bacteria? So again, this is just what I was saying before. If you have 2 bacterial cells here, if you have 2 bacterial cells here, and one just has normal chromosomal DNA, none of your plasmid, and the other one has actually managed to take up the plasmid, and this one has the, this is the plasmid with our, let's say it's got our ampicillin resistance gene in it. Then what we do is we expose all of your bacteria, including the ones with the plasmid and the ones that haven't, because it says in the question up here that the uptake of DNA by the bacteria is inefficient, so not all of the cells by any means are going to have the plasmid in them. What we can do is we can expose them with to ampicillin, and only the ones that have the plasmid will survive. The other ones will all die, so it selects for the bacteria with our plasmid, which is really useful for scientists because you can select just the ones that have successfully worked. What role does restriction enzymes have in nature? Well, restriction enzymes they're found in bacteria. And if you remember, when you have a bacteria like this, plasmid, there are these viruses that can come in called bacteriophages. I can't really draw them, and they attach to the outside and inject in some of their own viral DNA. And what these, so you have your viral DNA, which looks like this, and what the restriction endonucleases do is they come in and they will chop up the viral DNA. So they destroy, or they destroy / chop up viral DNA, protecting the bacteria from bacteriophages, nasty viruses. So you might think, well, you've got your nice plasmids and your chromosomal DNA and our bacteria. Why doesn't that get destroyed? And it has these proteins that protect it. The bacteria have co-evolved with these restriction enzymes, so that the actual DNA that the bacteria needs is not affected by them. They're protected. But these viruses, they are chopped up very quickly, and we've isolated them from these bacteria as scientists, and now we use them to chop up DNA for us, and they're very, very useful. So 6, it says, why might the enzymes work best at 37 degrees? And why should the unused enzymes then be placed in a freezer? And they work best at 37 degrees because the bacteria that they're isolated from, so the bacteria they were isolated from came from, or the bacteria they're isolated from usually had a home on humans, so infected or lived in humans. So they'd evolved to work best at human body temperature. And why are they placed in a freezer? Well, if you put them in a freezer, they freeze them, reduces their activity, and it prolongs their lifespan, so it keeps them fresh for us to be able to use. It prolongs their, let's say, useful life. Okay, so it says in step 4 you're asked to set up 2 tubes without restriction enzymes, so you'd have your plasmid and no restriction enzymes. Why are they important? Well, they're control tubes. Okay, they're control experiments. And what it means is if you had a gel and you loaded in your, this is for gel electrophoresis, so we have our wells here, and you loaded in your K+, which had our plasmid and our restriction enzymes, and our K-. This was the positive end here, and negative. And DNA is really negatively charged because of the phosphate groups on the backbone, so it will move towards the positive. So our K+, which we would think we would have a band that was quite large here, hasn't gone very far, very thick, and a small band because we've got 2 fragments. We've got the fragment that was the backbone of it that looked like that, and we've got the fragment that we cut out here with our restriction enzymes. So if we have that, our 2 fragments for the K+, and then we have our K-, the K- should just have the plasmid in it, so it should give us a very large band here. So it allows us to compare to make sure that we've got the products that we thought. If we didn't have that, we could just have the K+, and it could give us a band pattern which wouldn't tell us anything. So we need to compare it to the K- that just has the plasmid in. So that's why you would do that. Okay. Using your understanding of evolution, why would bacteria retain a gene that gives them resistance to antibiotics? How is the existence of bacterial antibiotic resistance affecting medicine today? This is very easy. It's useful to the bacteria because it improves their evolutionary fitness. It increases their survival rate. Thus improving evolutionary fitness. Why is it worrying? Because antibiotic resistance is spreading rapidly, and we've got some like we have things like MRSA, which is methicillin resistant Staphylococcus aureus, which has developed resistant to loads of antibiotics, and it's a real problem if we can't kill them with antibiotics because they're resistant. It will be an increase in human infections that are fatal. Okay. Bacteria, sea anemones, and humans seem on the surface to be very different organisms. Explain how a gene from a human or a sea anemone can be expressed in the bacteria to make a product never before made in a bacteria. Well, the DNA code is completely universal between those organisms, so that can be transferred. And transcription and translation are the same in all organisms. So DNA is universal. Transcription and translation are universal as well. Now, interestingly, bacterial RNA polymerase will only recognize bacterial promoters. So as long as you put in a promoter from a bacteria first, before your gene, it will express anything as long as that's a bacterial promoter. So in our case, we added the P-bad promoter, which is a bacterial promoter, which will allow it to attract bacterial RNA polymerase. So as long as you stick in one of those promoters first, before the gene, it doesn't matter where you put the gene in from, whether it's a sea anemone, whether it's a human, the bacteria will express it because it will think that the gene is, it's got a promoter that's from a bacteria, so it will think, oh, this must be a bacterial gene, and it will just start making it. So that's why that happens. Last question. Due to a mishap in the lab, bacteria carrying a plasmid with kanamycin resistance and bacteria carrying a plasmid with ampicillin resistance were accidentally mixed together. Describe an experiment that will allow you to sort out the two kinds of bacteria. Hint, make sure that you do not kill off one of the two kinds of bacteria you're trying to sort out. So they've both got resistance. So you would, if you don't want to, you don't want to kill all of them off, then what you need to do is you have your sample. Let's say it's in a little tube over here. This is the sample that's mixed up. You put it onto an agar plate, but take a small amount out. And this plate you're going to put some kanamycin on. Okay, so this is a kanamycin plate, as well as all the other stuff it needs to grow bacteria, but it has got this antibiotic in it. When you grow that, the only bacteria that will survive are your ones that are kanamycin resistant. So these are the strain that are kanamycin resistant. Okay. Okay. So then what you can do is you can take one of those kanamycin resistant bacteria and you can grow it on, and then it's pure again. It's not mixed with the other ones. And then what you'd need to do, just excuse me a second. Then what you would need to do is you would grab another one of the first set of sample, and this time you would grow it on a plate with the other antibiotic, which is ampicillin. So this is the ampicillin plate. And now only bacteria with ampicillin resistance grow, and again now you've sorted them out. Okay, hope that's been helpful.